section. Here we go. So this is looking at assembly, but before we do that, this is a thing that belongs really at the end of the previous section, but let's go see it anyway. So this is the poliovirus 3CD protease and polymerase. So the 3C, I believe, is this blue little thing that sits out here. That's a very protease fold, and I think the active site is right between these two little barrels. The polymerase is this thing, and so you've got your, let's see, it'd be nice if we could rotate it, but I believe you've got your palm at the bottom, I believe you've got your fingers over here, and I believe your thumb is this yellow bit that kind of reaches over. So this sort of flat bit down here would be the palm. And this is showing the way this thing moves as it is working. So it kind of jiggles around. It doesn't really open and shut the way I've described before, as far as we can tell. This is a simulation. But yeah, it's capable of a lot of movement. So whenever you see these proteins, don't assume that they are rigid structures. Assume that they have a little flexibility in there. Yeah. Okay, now on to assembly. So assembly is easy as VP1, VP2, VP3. All right. So VP1, VP2, and this VP3 over here come together, and they make a thing called a protomer, and it weighs 5 Svedberg units. So fantastic. You get 5 protomers together with that VP1 sticking up, and then boop, 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 boop. They're going to make that little VP1 star, and you've made a pentamer, which is this thing that I'm tracing right now on your screen. So that's a pentamer of 5 protomers. So there'd be 1, there'd be 2, there'd be 3, there'd be 4, and there'd be 5. And once you have the pentamers, you can put 12 of these pentamers together, and they will make the empty capsid. So the empty capsid weighs about 80 S. And after we assemble the entire empty capsid all by itself, then somehow, mysteriously, the genome gets loaded in there. So this is where we think the 2C is going to come in, that helicase-like enzyme. We think that may be the loader, because there are some bacteriophages where there is a helicase that is the loader protein. And so that's like a biological strategy that works for other viruses, and so that might be what we're doing. And that would be a good explanation, because we know that the 2C protein is absolutely essential. You cannot mess with that, or else the virus is just dead. And they're not sure why, because anything you would do to try and find out, you know, why could it be dead? It's just dead, flat out dead. So yeah, that's a tough one. Anyway, then the last stage, after we put together this capsid and loaded it up with the genome, we're going to cleave that last little bond between VP2 and VP4, and that's going to be done by a protease inside the cell. So that's the only cleavage that's not done by a viral protease. And then bam, you have your entire virion. It's 160S, and that's the one that's going to be released from the cell. Now, a friend of mine over in England showed that sometimes it gets released as VP0, so VP2 and 4 together, as if that cut didn't happen. And you know what? It doesn't make a lot of difference. The virus can still infect to some extent. So there we go. All right, so where do picornaviruses come from? Good question. So this is from a really cool paper that talks about the Big Bang of picornavirus evolution. It's by a guy called Eugene Koonin, who likes to think about these deep evolutionary problems. And so the idea here is that, yeah, basically everything that is a eukaryote has picorna-like viruses, picornavirus and friends. And the viruses, at least that were known at the time in plants, seem to be all related to each other. And the viruses in animals seem to be related to each other, like similar viruses in similar creatures. And so the thought was that, what if picornaviruses get into the common ancestor of all life and then spread out with that? So as plants differ from animals, then the picornaviruses of plants keep going with that, and they sort of merge, they diverge from the other picornaviruses and keep matching their host. Neat little theory. I think ultimately it's probably wrong, but there is a lot of interesting stuff in here. So let's see. Picornaviruses in general, or picorna-like viruses, all these things that are similar to picornavirus, have a couple of characteristics, and they're all arranged in different configurations. So there's always a VPG, and so that's shown as a little g or a little lowercase g, so that's the protein on there, and the actual protein that's part of the genome is shown as a G. So that's like the big square for the gene and the circle for the protein. And so that's on all of them except for a couple. There's a couple where it doesn't seem to be there, but in those cases we don't really know what's going on at the beginning, and so it may well be that those viruses have figured out another way to make their primer, or a way to make their primer and then cut it off from that protein, something like that. All right, they all have a 3C protease, so there you can see it's this sort of yellowy one, and the 3C protease, look at it, is always right before the RNA polymerase. So protease, polymerase, protease, polymerase, the polymerase is the RDRP. And so yeah, just over and over and over again, that seems to be the case, except in these couple of weirdos that seem like they're really just a couple genes from a Picornavirus that have gone off on their own and whatever. They're forming their own band. They're a solo act now. Yeah, a 3C-like protease, a 3D-like polymerase, and a VPG seems to be some of the big things. And then the other big feature that's included in most but not all of these is a helicase-like protein that's similar to 2C. So that's going to be the purple one. So S2H, S3H, and S3H up here, yeah. And so there's a few lineages that don't seem to have it, but most of the lineages do have it. So it's probably there in the common ancestor. Okay, so yeah, how do we fight back against Picornaviruses? Wow, yeah. We make a whole lot of microRNAs, and these are tiny little pieces of RNA. The average one's about 22 nucleotides long, and they are made by chopping down larger pieces of RNA. So you make a big long piece, then you chop it down to 22, and then that goes off and does something. MicroRNAs do not code for protein. They just work by sticking to the virus RNA and covering over certain parts that the virus needs. So for example, they can cover the start codon so it can't be translated. Or, for example, they could cover something like the CRE so that you can't make primers, which would be a good idea. The other way that they can work is by base pairing with the Picornavirus genome, and that will induce a thing called the miRNA, microRNA-induced silencing complex, the RISC. RISC. There we go. And that's going to lead to shredding the Picornavirus RNA and keeping the little 22 nucleotide microRNA. So that's kind of neat. And so over here on the side, this is a little summary of some of the microRNAs that are very important in Picornavirus. So these are all microRNAs that block Picornavirus replication by binding to different parts of the genome that the virus really needs. These are all microRNAs that block Picornavirus translation by binding to probably parts of the IRES so that they don't work the way they need to. They don't bind the ribosome just right. So yeah, it's neat. Our cells have been dealing with these stupid little Picornaviruses for long enough that we've actually built in a lot of defenses, even at the genetic level. You're a little bit genetically resistant to some Picornaviruses, so pretty cool. Okay, so in summary here, this is poliovirus. This is going to be our model Picornavirus. We'll pretend they all work exactly the same, even though we know they're all going to be slightly different. So positive strand RNA virus with a poly-A tail, but no cap. Instead of a cap, it has a VPG. Icosahedral capsid, that means a 20-sided capsid with 12 five-fold symmetry axes, 12 of those little star-shaped things made by VP1. Each of those VP1s has a little canyon, or rather, there's a little canyon in between them for receptor binding, so that's where they're going to attach to the receptor. They all replicate in the cytoplasm, so they don't need to go into the nucleus. And remember that flu needed to go into the nucleus and HIV needed to go into the nucleus, but corona didn't need it, and poliovirus does not need it. So polio and corona are kind of similar. It causes the cell to stop being able to make any proteins at all by cutting EIF4G and then replacing it with an IRES on its own RNA that is not present on the cell's RNA. So yeah, it breaks something else so you can't have it. Nice. It's going to move a lot of host cell proteins around inside the cell, and so we didn't cover a whole lot of this, but poliovirus thoroughly rearranges the cell to its own liking. It is very, very good. A little bit OCD, maybe. Yeah. And then the polyprotein is going to be translated. So we'll make that one giant protein, and it's going to be processed, which means cut into pieces by the viral protease, which you'll remember is the 3C protease. And then it's going to replicate via a double-stranded RNA intermediate, which means it's going to take a positive strand, make a negative strand, and so the positive and negative together make a little double-stranded RNA, neat. And then it's going to take a negative strand and make a bunch of positive strands, and so once again it's kind of double-stranded for a minute while it's making that copy, and then it peels off that copy and makes another one. Yeah. And so it requires a lot of host cell factors, like a constant supply of proteins from the cell to keep replication going. And we didn't go into those. There's a lot more detail on all of these areas. I just tried to pick out a couple of highlights for you. Lastly, the VPG, once we've added 2 uridines to that, so VPG with 2 U's on it, becomes the primer for RNA synthesis by the 3D polymerase. And then when we're getting ready to translate this and bind that IRES on there, we'll actually wait for the cell to cleave off the protein from the RNA. And the cell does this automatically because that's a weird thing, this protein bound to the RNA, and so cells just naturally, boom, snap it. And that allows the IRES to interact with the ribosome in the right way to cause translation. So there you go. That's the end of that.